Hey guys, welcome back. Today's topic is excretion in human beings. This is from the chapter Life Processes of Class 10 Biology. Let's begin. Metabolic activities generate nitrogenous materials which need to be removed. These nitrogenous waste materials, if they accumulate inside the body, they become toxic. So they need to be removed from the body. The biological process involved in the removal of these harmful metabolic waste from the body is called excretion. Okay, so this is what the biological process involved in the removal of these harmful metabolic wastes from the body is excretion. Okay, now moving to the continuation here, ammonia, urea and uric acid are the major forms of nitrogenous waste excreted by the animals. Okay, this is very important. Okay, I repeat this statement once again. Ammonia, urea and uric acid are the major forms of nitrogenous waste materials that are excreted by the animals. Okay, here ammonia is most toxic and require a large amount of water for its elimination. Okay, so this is important for exams. Okay, so ammonia is the most toxic form and require large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. So among these three major forms of nitrogenous waste, this ammonia is most toxic and uric acid is least toxic. Okay, and for the removal of ammonia, large amount of water is needed, and for the removal of uric acid, least amount of water is needed. Okay, now moving to the continuation, the process of excreting ammonia is called ammonotelism. Okay, so you can remember this word ammonotelism, very important word. Okay, so it is nothing but the process of excreting ammonia. Ammonia is excreted, that is ammonotelism. Okay, many bony fishes, aquatic amphibians and aquatic insects are ammonotelic in nature. These organisms, okay, many bony fishes, aquatic amphibians and aquatic insects, they are ammonotelic. It means they excrete ammonia as nitrogenous waste material. Okay, ammonia is actually readily soluble. Okay, it is readily soluble. Okay, generally it is excreted by diffusion across the body surface. Okay, ammonia is readily soluble and it is excreted by diffusion process. Okay, excreted by diffusion across body surfaces. Okay, or through gill surface, okay, in case of fishes as ammonium ions, okay. So I repeat this once again. Here ammonia is readily soluble in water and it is excreted by diffusion across the body surface or across the gill surfaces as ammonium ions. So this is how the ammonia is excreted, and the mechanism is called ammonotelism, which is found in many bony fishes, aquatic amphibians and aquatic insects. Okay, now the continuation part of this. Okay, mammals many terrestrial amphibians and marine fishes mainly excrete urea. Okay, so here mammals, they have mammary glands, so they are called mammals. Okay, here many terrestrial amphibians. Here in the previous one we discussed, okay, these are aquatic amphibians are excreting ammonia, but the terrestrial amphibians, they are excreting urea. Okay, and marine fishes, okay, these marine fishes also excrete urea, and as they excrete urea, this mechanism is called ureotelic animals, and the mechanism is ureotelism. Okay, so the mechanism is ureotelism, and these animals are called ureotelic animals. Examples are mammals, terrestrial amphibians, and marine fishes. They excrete urea. Okay, ammonia produced by metabolism is converted into urea in the liver of these animals. So in these animals, the ammonia which is produced during the metabolic reactions, it is converted into urea during the metabolism in liver. Okay, so this is what happens. The urea is formed and it is excreted through the kidneys, but certain amount of this, okay, certain amount of urea is retained in kidney. Okay, it is retained in kidney matrix, okay, of some of these animals to maintain desired osmolarity, to maintain desired osmolarity. Okay, so this is again one important concept, okay, you can remember. Generally urea is excreted by the kidneys, but certain amount of urea is it is retained in the kidney matrix. This is very much helpful in maintaining the desired osmolarity. Okay, now the continuation part of this: reptiles, birds, land snails, and insects, okay, they excrete nitrogenous waste as uric acid, okay, in the form of pellet or paste with a minimum loss of water, and these animals are called uricotelic animals. Okay, ureotelism is the mechanism. The animals are called uricotelic animals, and here the excretory product here is uric acid. Okay, and the reptiles, birds, land snails, and insects excrete uric acid either in the form of pellet or in the form of paste with a minimum loss of water. So this mechanism is called ureotelism. Okay, so what are the major forms of nitrogenous waste excreted by animals? The answer is ammonia, urea, and uric acid. If ammonia is excreted, then the mechanism is called ammonotelism. If urea is excreted, then the mechanism is called ureotelism. If uric acid is excreted, then the mechanism is called ureotelism. Okay, now after this, the continuation part. Okay, many unicellular organisms remove these wastes by simple diffusion. Okay, so many unicellular organisms, single celled organisms, they remove this nitrogenous waste by simple diffusion from the body surface into the surrounding water. Okay, and complex multicellular organisms use specialized organs to perform the same function. So these specialized organs are formed because of differentiation process. Okay, so this differentiation is the process by which living organisms, living cells attain a definite shape, size, and perform specific functions. So in complex multicellular organisms, okay, specialized organs perform the function of excretion. Okay, so here different organs use the varied strategies to remove the metabolic waste from their body, and these are the mechanisms. Now, continuation of this, the excretory system. Now we are going to discuss in detail about excretion in human beings. Okay, the excretory system of human beings include a pair of kidneys, a pair of ureters, a urinary bladder, and urethra. So these four structures, these are very important in excretory system. You can see here, you can see the one, this one is the kidney, okay, then ureter, then urinary bladder, then urethra. So these are, okay, the parts in human excretory system. So the excretory system in humans includes a pair of kidneys. You can see this, these are the two kidneys, a pair of kidneys, okay, and then a pair of ureters. You can see here, this is left one, this is right. So a pair of ureters. After that, urinary bladder and urethra. You can see here, this is urinary bladder and this one is urethra. Okay, so these are the parts in human excretory system. Now here, these kidneys are located in the abdomen. Okay, kidneys are located in the abdomen, one on either side, one on either side of the backbone. Okay, so this is the location of kidneys in humans. Okay, I repeat this once again. Here, human kidneys are located in the abdomen, and these kidneys are one on either side of the backbone. This is the left kidney and this one is the right kidney. Okay, you can see the position. What difference do you see here in the height, here in the position? What difference do you see? Okay, this right side, the kidney is slightly lower. Can you guess what is the reason behind this? Why this right kidney is lower in position when compared with this left one? You can see if we plot a straight line, okay, so this one is a little bit in higher position when compared with this. The reason behind this is there is a largest gland in the body called liver on right side. Okay, so that is the reason right kidney is slightly lower than the left kidney. One of the important concept for exams. So what is the position of kidneys? They are in the abdomen, one on either side of the backbone. Okay, if you see this picture, the other structures on the top of the kidney, this cap like structure is adrenal gland. Okay, and after that you can see this one is a renal artery. Okay, renal artery supplies oxygenated blood to the kidney. Okay, and renal vein, the branch of this vena cava, renal vein, carries deoxygenated blood from kidneys. Okay, so these are the parts, and in a kidney structure, the outer part is called cortex and the inner part is called medulla. You can see here, this outer surface, this outer part, outer region is cortex, and the inner part, this one is the medulla. Okay, and you will see here a funnel like structure, okay, into which these ureters are seen. Here ureters are arising from this. Okay, a funnel like structure you can see, that is what we call pelvis. Okay, and this is inferior vena cava, and this one is aorta, and this is a branch of aorta, renal artery. Okay, so this is about human excretory system, the different parts in it. Okay, now we will discuss what are the functions of these different parts in the human excretory system. Here, urine produced in kidneys. Okay, so production of urine occurs in kidneys. So urine produced in kidneys passes through the ureters into urinary bladder, where it is stored until it is released through the urethra. Okay, so here the urine production occurs. After that, that urine is carried through this ureter, okay, to the urinary bladder, where it is temporarily stored. After that, it is passed out of the body through this urethra. So kidneys, urine formation occurs. Ureters carry the urine from kidneys to urinary bladder. Urinary bladder temporarily store urine, and the urethra is the opening through which, okay, here you can see through this urethra urine is passed out of the body. Okay, now after this, the purpose of making urine is to filter out waste products from the blood. Okay, as we discussed in the beginning, okay, because of metabolic reactions, several nitrogenous wastes are formed in the living cells, and those living cells are present in the those metabolic wastes are present in the blood. Okay, so here to filter out these waste products from the blood, that is the main purpose of making urine, and this is the primary function of kidneys, the excretory organs in humans. Okay, now here, just as CO2 is removed from the blood in lungs, okay, so similarly nitrogenous wastes like urea, okay, uric acid, okay, are removed from blood, okay, in the kidneys. Okay, so this is one small comparison between the functioning of lungs and the functioning of kidneys. Okay, here CO2 is removed from the blood in the lungs. CO2 is the waste product here. Okay, similarly nitrogenous waste like urea, uric acid are removed from blood in the kidneys. Okay, here CO2 is removed from the blood in lungs, and in this case urea, uric acid, the nitrogenous wastes are removed from the blood in kidneys. So this is a comparison between the functioning of lungs and the functioning of kidneys, okay, with respect to their waste products. Okay, here the basic filtration unit in the kidney is a cluster of very thin walled blood capillaries. I repeat this once again. Okay, a basic filtration unit in the kidneys is a cluster of very thin walled blood capillaries that collects the filtrate. Okay, so here a cluster of very thin walled blood capillaries collects the filtrate. Okay, and here this filtration unit is nothing but the nephron. Okay, and it is a cluster of very thin walled blood capillaries. Each capillary cluster in the kidney is associated with the cup shaped end called coiled tube called Bowman's capsule. Okay, I repeat this once again. Each capillary cluster in the kidney is associated with a cup shaped end of a coiled tube called Bowman's capsule. Okay, you will find here in a diagram, you will understand better. You can see this is a cup like Bowman's capsule. Okay, you can see here this is the Bowman's capsule. This part is a Bowman's capsule, the cup like structure. Okay, so here each capillary cluster in the kidney is associated with this Bowman's capsule. You can see this is a capillary cluster, and this is what we call glomerulus. Okay, it is a tuft of capillaries. This is a cluster of capillaries, and this glomerulus is associated with a cup-like structure called Bowman's capsule. Okay, so this is one of the very important aspect. Okay, this tuft of capillaries is called glomerulus, and this cup-like structure of the tubule is called Bowman's capsule. Okay, so this is about the association between glomerulus and Bowman's capsule. Okay, each kidney has large numbers of these filtration units called nephrons. Placed, they are packed close together. Okay, so this one is a nephron, and large number of nephrons are present in each kidney, and they are packed closely. Okay, so they are packed close together. Okay, some substances in the initial filtrate, okay, such as glucose, amino acids, salts, and major amount of water are actively reabsorbed as the urine flows along the tube. Okay, so as the urine flows along this tube, okay, so here these substances like glucose, amino acids, salts, and large amount of water actively reabsorbed. Okay, so this is one of the very important step in the mechanism of urine formation. Okay, and here if we look, there are three steps in the mechanism of urine formation. One is glomerular filtration. One is glomerular filtration. Okay, the second step is tubular reabsorption. Okay, and the third step is tubular secretion. Okay, so these are the three important steps in the mechanism of urine formation. Okay, the first step is glomerular filtration. You can see this is a tuft of capillaries called glomerulus. Okay, and this one is a Bowman's capsule. Okay, and this is afferent renal arteriole. It is supplying the blood to here. It is carrying the blood here into the nephron. Okay, now here the blood is filtered. Okay, you can see here the blood is filtered through these capillary walls, the basement membrane present between the capillary walls, and here the Bowman's capsule. Okay, so Bowman's capsule is also made up of single cell thick squamous epithelium. So Bowman's capsule, capillary walls, basement membrane between them. Through these layers, the blood is filtered into this cup-like structure. Okay, so into the tubular part, the filtrate is entering. So this is what we call the first step, glomerular filtration. Okay, it means the blood is filtered. Okay, now the second step is tubular reabsorption. Okay, this is nothing but while the filtrate is carried in this tube, okay, while the filtrate is passing through this tube, the useful substances present in this filtrate, as I discussed just now, glucose, okay, amino acids, okay, here salts, okay, and large amount of water, all these useful substances, okay, large amount of these useful substances actively absorbed. This is what we call tubular reabsorption. Okay, after the reabsorption step, the next one is tubular secretion. So excess ions like hydrogen ions, potassium ions, like this, excess ions are secreted by the tubular cells here into the filtrate. So these ions are secreted into the filtrate by the tubular cells. Okay, so this is the third step, and as a result the urine is formed. So these are the three important steps in the mechanism of urine formation. Okay, now here the important aspect is substances like glucose, amino acids, salts, major amount of water are selectively, okay, they are reabsorbed as the urine flows along the tube. Okay, the selective reabsorption take place. Okay, now after this the amount of water reabsorbed depends on the two aspects. One is how much excess water there is in the body. Okay, how much excess water is present in the body, that is deciding, okay, the amount of water reabsorbed. Okay, and also how much dissolved waste there is to be excreted. Okay, so the two aspects, how much excess water is present in the body and how much dissolved waste there is to be excreted. So these two factors will decide how much amount of water is reabsorbed. Okay, so this is one very important aspect for exams. Okay, now after this, moving to the continuation, the urine forming in each kidney eventually, okay, enters a long tube, the ureter. Okay, the urine which is formed in each kidney eventually enters into a long tube, that is what we call ureter, which connects the kidneys with urinary bladder. Here you can see the picture. Okay, in this picture you will find, okay, how the ureter is connecting the kidney with urinary bladder. Okay, so this structure is a ureter, this one. Okay, it is connecting the kidney and urinary bladder. The urine which is produced here is carried to the urinary bladder through this ureter. Okay, so the urine which is formed in kidneys is entering into the long tubes, ureters, that are connecting kidneys with urinary bladder. Okay, now after this, the continuation part of this. Okay, urine is stored in urinary bladder until the pressure of the expanded bladder leads to the urge to pass it out through the urethra. Okay, so the pressure of the expanded bladder, when the urine accumulates, okay, when the urine accumulates inside this bladder, okay, then the pressure is created, and this, because of that pressure, this bladder will expand. Okay, and that develops an urge to pass the urine out of the body. Okay, so this is how, okay, an urge is developed. Okay, the urge to pass the urine out through the urethra, that is created because of the pressure of the expanded bladder. Okay, the bladder, the urinary bladder is muscular. Okay, it is muscular and it is under neural control. Okay, the urinary bladder is muscular and it is under the neural control. As a result, we can usually control the urge to urinate. Okay, I repeat this once again. The urinary bladder is muscular and it is under neural control. So, we can usually control the urge to urinate. Okay, so this is about excretion in human beings. Now, one very important concept in connection with this is artificial kidney. Okay, and here the mechanism that we are going to discuss is about, okay, hemodialysis. Okay, here several factors like infections, injury or restricted blood flow to kidneys reduces the activity of the kidneys. Okay, so these are the reasons. Okay, infections, injury or restricted blood flow to the kidneys reduce the activity of the kidneys. Okay, and this leads to accumulation of poisonous waste in the body, which can even lead to death. Okay, so because of these reasons, the activity of the kidneys is reduced, and because of that, the accumulation of poisonous waste, okay, the nitrogenous waste occur in the body, and in severe conditions it can lead to death. Okay, kidneys are the vital organs for survival, but because of these reasons their activity can reduce, and in severe conditions it may lead to death. Okay, now here, in case of kidney failure, an artificial kidney can be used, and this artificial kidney is a device to remove nitrogenous waste product from the blood through dialysis process. Okay, I repeat this once again. This is very important statement. Okay, in artificial kidney, okay, it is a device to remove. Okay, I repeat this once. It is a device to remove nitrogenous waste product from the blood through dialysis. Okay, so here you can see the picture. Okay, this is what we call hemodialysis. Okay, here the blood line from the artery to pump. Okay, so here the blood is taken from the patient, and with the help of tubes, okay, with the help of pump, now it is passed into this hemodialysis machine. Okay, that is artificial kidney. Okay, and now the nitrogenous waste like urea present in this blood will diffuse into the dialyzing fluid which is present in the chamber. Okay, and hence the blood is purified. After that, the purified blood is injected back into the patient. Okay, and this dialyzing fluid is added freshly after certain duration. Okay, so here you can see artificial kidneys contain a number of tubes with semi-permeable lining suspended in a tank filled with dialyzing fluid. You can see here, this is a tank filled with dialyzing fluid. See, this is a fresh dialyzing fluid. Okay, this tank is filled with fresh dialyzing fluid. Okay, now you can see artificial kidneys contain a number of tubes with semi-permeable lining. Okay, so these tubes have semi-permeable lining. Okay, it allows the selected molecules to pass through them. Okay, so these tubes have semi-permeable lining. Okay, so now here this fluid has the same osmotic pressure as blood, except that it is devoid of nitrogenous waste. Okay, so the difference between the dialyzing fluid and the blood, okay, is, okay, here the nitrogenous waste are absent. Okay, so this fluid has the same osmotic pressure as blood, except that it is devoid of nitrogenous waste. So here this dialyzing fluid do not have nitrogenous waste. The blood taken from the patient has nitrogenous waste. So as the blood is passing through these tubes, through the semi-permeable membranes present in the tubes, the nitrogenous waste can diffuse out into, okay, this dialyzing fluid, and they are removed. Okay, so this is how the nitrogenous waste, they are moving from their higher concentration to lower concentration, and they are removed from the blood, okay, into dialyzing fluid, and the used dialyzing fluid is removed frequently. Okay, now the continuation part. The patient's blood is passed through these tubes. During this passage, the waste products from the blood pass into dialyzing fluid by diffusion. Okay, this is what we call dialysis. Okay, the patient's blood is passed through these tubes. During this passage, the waste products from this blood pass into dialyzing fluid by diffusion. Okay, and this purified blood is pumped back into the patient. Okay, this is similar to the functioning of kidney. This is similar to the functioning of kidney. Okay, this is similar to the functioning of kidney, but it is different since there is no reabsorption. No reabsorption involved. Okay, so this is different. Though the artificial kidney is similar to the functioning of kidney, the difference here is no reabsorption involved. Okay, so this is how the blood is purified. Okay, normally in a healthy adult, the initial filtrate in the kidneys is about 180 liters daily. Okay, very important question for exams. Okay, so what is the amount of initial filtrate formed in one day? The answer is 180 liters. Okay, so in normal conditions, in a healthy adult, the initial filtrate in the kidneys is about 180 liters daily. Okay, however, the volume actually excreted is only a liter or 2 a day. It means 1 to 2 liters of urine is actually excreted. Okay, and because the remaining filtrate is reabsorbed in the kidney tubules. Okay, so this is how, though the initial filtrate formed is 180 liters daily, only 1 to 2 liters of filtrate, the urine is excreted, remaining all is reabsorbed in the kidney tubules. Okay, so this is how excretion process occurs in human. Okay, in humans, urea is the major nitrogenous waste. Okay, the nitrogenous waste is urea. So we find ureotelism in humans. Okay, now here the major forms of nitrogenous waste are ammonia, urea, uric acid. Among these three, ammonia is most toxic, uric acid is least toxic. Okay, human excretory system consists of a pair of kidneys, a pair of ureters, urinary bladder and urethra. Okay, inside the kidneys, urine production occurs. Ureters are transferring the urine from kidneys into the bladder, and the urinary bladder stores the urine temporarily, and through urethra the urine is excreted out. Okay, in the mechanism of urine formation we find three steps. First, glomerular filtration, second, tubular reabsorption, and the third, tubular secretion. The initial filtrate formed is 180 liters and the urine excreted is 1 to 2 liters daily. Okay, so this is all about today's session. If you are new to my channel, subscribe for more sessions. If you liked it, share it with your friends. I'll be back with more sessions. Have a